Hi, and welcome back to Epidemics and the Evolution of Medicine. This is our last week together. Originally, I had planned to study just Ebola because of my almost involvement with last year's epidemic, as I discussed in my original post for this course. But with the recent MERS outbreak, I decided to broaden the focus to include MERS and SARS because they all represent new viruses that have emerged as threats over the last several years. This is the new face of infectious disease. To begin, each of these viruses, like the H1N1 influenza virus and the human immunodeficiency virus, are zoonotic, meaning that they are viruses that began in animals and jumped to humans. And like H1N1 and HIV, they can cause devastating disease that can spread rapidly because of contemporary behavior. Finally, like H1N1 and HIV, they have proven to be exceptionally challenging to scientists and physicians. We will begin with a brief history of each virus. Ebola, also known as Ebola hemorrhagic fever, is a disease caused by one of 5 different Ebola viruses, 4 of which can cause severe illness in humans and animals. Ebola is a member of the filovirus family, and it's shown here, an electromicrograph is shown here on this slide. The first human outbreaks occurred in 1976, one in Zaire in Central Africa and the other in southern Sudan. The virus is named after the Ebola River, where the disease was first described. The incubation period of Ebola virus disease ranges from 2 to 21 days. The onset of illness is sudden, with fever, headache, joint and muscle pain, sore throat, and intense weakness. This is then followed by diarrhea, vomiting, rash, impaired kidney and liver function, and stomach pain. Some patients may develop a rash, red eyes, hiccups, internal and external bleeding. Ebola hemorrhagic fever is fatal in between 50 to 90% of all clinically ill cases. Ebola is extremely infectious, but not extremely contagious. It is infectious because an infinitesimally small amount can cause illness. Laboratory experiments on non-human primates suggest that even a single virus may be enough to trigger a fatal infection. The first human case in an outbreak of Ebola is acquired through contact with blood, secretions, organs, or other bodily fluids of an infected animal, and this is thought to often occur with butchering of an animal for food. Scientists now believe that bats are the natural reservoir for the virus, and that apes and humans catch it from eating food that bats have drooled or defecated on. Such a lovely thought. Or by coming in contact with surfaces covered in infected bat droppings and then touching their eyes or mouths. The virus is then transmitted to others through direct contact with the blood, secretions, organs, or other bodily fluids of infected persons. Typically, symptoms appear 8 to 10 days after exposure. People can also become infected through contact with objects, such as needles or soiled clothing that have been contaminated with infected secretions. As in the recent epidemic, hospital workers have frequently been infected in Ebola outbreaks through close contact with infected patients and insufficient use of correct infection control precautions and barrier nursing procedures. Outbreaks have also been fueled by traditional burial practices in which mourners have direct contact with the bodies of the deceased. Acquisition via sexual contact with a convalescent case is theoretically possible, as the virus is present in semen for up to 3 months after recovery, although survivors are not usually considered to be infectious. Ebola virus is considered only moderately contagious because it is not spread through routine social contact, such as shaking hands or sitting next to someone, with asymptomatic individuals, and there is no evidence of transmission through intact skin or airborne spread through droplets with coughing or sneezing. Laboratory diagnosis must be carried out under maximum biological secure conditions. Tests are available to detect viral RNA or antibodies to the virus, and it may also be isolated in cell culture. These tests have been developed to allow rapid lab diagnosis during outbreaks. As the goal of interrupting chains of Ebola virus transmission depends heavily on lab support, the WHO has stimulated the development of rapid, safe, and reliable point-of-care diagnostic tests. Containment efforts have been slowed by cumbersome, time-consuming, and complex diagnostic tests that, while highly accurate, impose a number of additional logistical challenges, including transportation needs and requirements for a high level of lab safety. Delays in receiving test results are especially detrimental to outbreak control. Apart from posing a severe risk to families and communities, undiagnosed and unmanaged patients contribute to the cyclical transmission pattern currently being seen, whereby cases begin to fall as control measures take effect, only to spike again as new chains of transmission are ignited. The inability to get rapid diagnostic results can mean that uninfected people are placed together with confirmed cases in transit centers where the risk of infection is high. Better diagnostic tests make patient triage quicker, safer, and most importantly, efficient. The rapid point-of-care diagnostic test will boost the Ebola response considerably, both now and later, as the number of cases declines and the strategy shifts to detecting and breaking the last chains of transmission. To avoid person-to-person transmission of Ebola virus, great care needs to be taken when nursing patients to avoid contact with infected bodily fluids. Patients should be isolated and strict barrier nursing techniques should be used, including wearing masks, gloves, and gowns. Invasive procedures such as the placing of intravenous lines, handling of blood, etc. are a particular risk, and strict infection control is essential. Non-disposable protective equipment must be properly disinfected before reuse. The bodies of those that have died of Ebola virus remain highly infectious and should be promptly and safely buried or cremated. And here you see on this slide a team of healthcare workers in their protective gear about to begin their shift. There is no specific treatment or vaccine available for Ebola virus disease, but potential new vaccines and drug therapies are being developed and tested. Patients require intensive supportive therapy, including intravenous fluids or oral rehydration with solutions including electrolytes maintaining their oxygen status and blood pressure. Selected cases, and this means Westerners and healthcare workers, were given antiviral agents like ZMapp, a combination of 3 antiviral antibodies, and transfusions from individuals who had recovered from Ebola. Both measures seem to improve outcomes if given early in the course of the illness, although the number of cases is too small to really make any statement. The recent epidemic began in December of 2013 in a small remote village in Guinea. A 2-year-old child became suddenly and severely ill with fever, diarrhea, and black stools, a sign of GI hemorrhage. He died 2 days later, followed by his mother and 2 sisters. Epidemiologic investigators have visited the village and speculate that the virus came from a small bat that is frequently used as food and lives in large numbers in the village under the roofs of the huts. Because diarrhea was the predominant symptom, Ebola was not suspected as the cause, even as illness spread through the village. Previous Ebola outbreaks occurred in Central Africa, which is much less populated than West Africa, where Guinea is located. In remote, low-population areas, containing a viral outbreak is easier as infected patients can be kept isolated. All previous outbreaks have occurred in such low-population areas and have died out after at most 3 months, with a combination of isolation coupled with likely attenuation of the virus over time. In the current outbreak, the disease spread rapidly from one village to another without formal recognition as an Ebola outbreak for several months. By March of 2014, cases were reported in Liberia and Sierra Leone in people who had traveled to Guinea, and Médecins Sans Frontières, MSF, an international medical aid group, sent the first samples of what turned out to be Ebola to labs in Germany and France in March 2014. MSF teams were already on site, struggling to control the spread of the disease. By June, MSF was reporting that the epidemic was out of control. In July, cases began to be reported in other parts of Africa where patients had flown, and then in August, 2 American missionaries working with an international medical aid group were infected. Their emergency evacuation to Atlanta suddenly brought widespread attention and then panic as health officials and the general public realized that this disease, with its incredibly high mortality rate, had the potential to spread around the world. Both of the missionaries survived after receiving transfusions from Ebola survivors and antiviral medications. But on August 8, 2014, the World Health Organization officially declared the outbreak to be a global health emergency. By mid-August, there had been 2,000 known cases, of whom more than half had already died. September 2014 saw the beginning of an international governmental response, with the first official commitment from the U.S. of $500 million and 3,000 troops deployed to the area to deliver supplies and build new treatment centers. On September 30th, 2014, the first American who had traveled to the U.S. from Liberia was belatedly admitted to a Dallas hospital with a diagnosis of Ebola. He was the first American case of Ebola. Despite maximal care, this patient died, and 2 of the nurses who cared for him were infected. Although they both survived, these cases created a global public health crisis with widespread panic fueled by 24-hour news coverage. Through the fall of 2014, the epidemic continued, and multiple healthcare workers were emergently transported back to their home countries with Ebola. On the ground in West Africa, healthcare workers struggled in protective gear that was so confining they could only work 4-hour shifts. One nurse reported literally pouring sweat out of her rubber boots at the end of her short shift because it was so hot in the hazmat suit. Attempts were made by teams of international workers to identify and isolate all potential cases, despite continuing bed and facility inadequacies. In early October, the UK announced that it would introduce temperature screening for passengers arriving at Gatwick and Heathrow airports from West Africa, followed by a similar statement regarding JFK for the United States. On October 14th, WHO announced that the fatality rate from Ebola had reached 70%, with the total number of cases at 6,353 and deaths topping 4,447. Gradually, the number of new cases slowed, with both Liberia and Sierra Leone reporting the lowest number in 6 months during the first week of January 2015. On January 27th, the World Health Organization reported that confirmed cases exceeded 22,000 and deaths 8,800. On March 20th, Liberia recorded its first case of Ebola in more than 2 weeks, and by May, Liberia was reportedly Ebola-free, with continually declining cases in Sierra Leone and Guinea. In total, there were over 26,000 cases with more than 11,000 deaths. And this is a graph shows this from April to the following May. In April of 2015, the Centers for Disease Control and Prevention, in partnership with the Sierra Leone College of Medicine and the Sierra Leone Ministry of Health and Sanitation, announced the beginning of a vaccine trial against Ebola. The study is enrolling and vaccinating volunteers for the Sierra Leone trial to introduce a vaccine against Ebola, or STRIVE. This study will assess the safety and efficacy of the RVSV-ZBOV candidate Ebola vaccine among health and other frontline workers. STRIVE will enroll about 6,000 individuals in areas that were heavily infected during the most recent epidemic. When participants enroll in the study, they will be assigned randomly to one of 2 time frames for vaccination, either immediately or about 6 months later. All study participants will receive the vaccine and be followed closely for 6 months. The study will evaluate if and how well the vaccine works by comparing rates of Ebola virus disease in those who are vaccinated to those who have not yet received the vaccine. The ZBOV candidate vaccine uses a vesicular stomatitis virus carrying a non-infectious Ebola virus gene. The vaccine cannot cause Ebola, but can potentially stimulate an immune response to protect against the disease. The vaccine was developed by the Public Health Agency of Canada's National Microbiology Lab and licensed to New Link Genetics. The vaccine has and continues to be studied in hundreds of people. As of March 26, 2015, more than 800 people in Africa, Canada, Europe, and the United States. Results from early studies to date show an acceptable safety profile and indicate that the candidate vaccine produces an immune response. The Biomedical Advanced Research and Development Authority is supporting the advanced development and manufacturing of the vaccine and is assisting the CDC in conducting the clinical trial in Sierra Leone. Several other labs have reported development of potentially effective vaccines, but trials have not been initiated because of the cost and the reluctance of some pharmaceutical companies to be involved in what they view as a non-profitable venture. SARS, or the Severe Acute Respiratory Syndrome, is a viral respiratory disease which is zoonotic in origin, caused by the SARS coronavirus. SARS first appeared in China in November 2002. It is spread through airborne droplets, but may also be spread on contaminated objects like doorknobs, telephones, and elevator buttons. Symptoms of acute respiratory distress can progress rapidly to respiratory failure and death. Within weeks, SARS spread from Hong Kong to infect individuals in 37 countries in early 2003, and within a few months, cases were reported worldwide, carried by unsuspecting travelers. Ultimately, there were over 8,000 cases and 774 deaths reported in multiple countries, with the majority of cases in Hong Kong, where the fatality rate was just under 10%. SARS showed us how quickly infection can spread in a highly mobile and interconnected world. However, concerted international cooperation allowed health experts to quickly contain the spread of the disease. The epidemic appears to have started in Guangdong Province, China in November 2002. Chinese government officials did not inform the World Health Organization of the outbreak until February 2003, when an American businessman traveling from China developed a pneumonia-like illness while on a flight to Singapore. The plane stopped in Vietnam, where the victim subsequently died. Several of the medical staff who treated him soon developed the same disease despite basic hospital procedures. An Italian doctor, Carlo Urbani, identified the threat and communicated it to the World Health Organization and the Vietnamese government before he died. The disease spread in Hong Kong from a mainland doctor who arrived in February and stayed at a hotel in Kowloon, infecting 16 hotel visitors. Those visitors traveled to Canada, Singapore, Taiwan, and Vietnam, spreading SARS to those locations. With isolation and respiratory precautions under the direction of an international public health consortium, the pandemic was arrested in the summer of 2003. The CDC and Canada's National Microbiology Lab identified the SARS genome in April 2003. Scientists in the Netherlands then infected macaques with that virus, leading to development of the same symptoms seen in humans, thus fulfilling Koch's postulates and confirming that the SARS coronavirus was the causative agent. In 2005, 2 studies identified a number of SARS-like coronaviruses in China's Chinese bats. Phylogenetic analysis of these viruses indicated a high probability that SARS coronavirus originated in bats and spread to humans either directly or through animals like civets in Chinese markets. The bats did not show any visible signs of disease, but are considered to be the likely natural reservoirs of the virus. In late 2006, scientists from the Hong Kong University and the Guangzhou Center for Disease Control and Prevention established a genetic link between the SARS coronavirus, which appears in civets and in humans, proving that the disease had jumped across species. PCR testing can identify the virus in potentially infected individuals, but there have been no reported cases since 2004. MERS, or the Middle East Respiratory Syndrome, is caused by the Middle East Respiratory Syndrome coronavirus, a single-stranded RNA novel species of the genus Beta coronavirus. Initially called Novel Coronavirus 2012, it was first reported in 2012 after genome sequencing of a virus isolated from sputum samples from patients who fell ill in a flu outbreak. And on this slide you can see an electron micrograph of the MERS virus. Most people confirmed to have MERS have had a severe acute respiratory illness, but some have also had GI symptoms. For many people with MERS, more severe complications followed, such as pneumonia and kidney failure. While up to 40% of patients have died, some infected people have also had very minimal symptoms. Most fatal cases have occurred in individuals with an underlying medical condition. Some infected people had mild symptoms or no symptoms at all, and they recovered completely. All age groups have been infected. The incubation period for MERS is usually about 5 to 6 days, but it can range from 2 to 14 days. MERS, like other coronaviruses, is thought to spread from an infected person's respiratory secretions, such as through coughing. Person-to-person spread, usually after close contact, such as caring for or living with an infected person, has been well documented. Infected people have spread MERS to others in healthcare settings, such as hospitals, but there has so far been little community spread. The virus appears to have originated in bats, but serological evidence shows that these viruses have infected camels for at least 20 years, and to have spread from camels to humans in approximately the year 2010. The original bat host species and the time of initial infection in this species has yet to be determined. The coronavirus infection in dromedary camels has been shown to have 99.9% matching to the genomes of the human MERS virus. Camels are thought to serve as the primary source of the virus infecting humans, with bats as the ultimate reservoir. As of June 2014, there had been 689 cases of MERS reported within the Kingdom of Saudi Arabia, with 283 deaths. In June 2015, MERS cases had been reported in over 20 countries, including Saudi Arabia, Jordan, Qatar, Egypt, the United Arab Emirates, Kuwait, Turkey, Oman, Algeria, Bangladesh, Indonesia, Austria, the United Kingdom, South Korea, the U.S., mainland China, and Thailand. To this time, all reported cases have been linked to countries in and or near the Arabian Peninsula. Most infected people either lived in the Arabian Peninsula or recently traveled from there before they became ill. A few people became infected with MERS after having close contact with an infected person who had recently traveled from the Arabian Peninsula. Then in May of 2015, the virus appeared in South Korea. This slide shows where confirmed cases have occurred up until May. At that time, the virus appeared in a 68-year-old man who had just returned from a trip to Saudi Arabia. By June 18th, a total of 165 patients have been identified in South Korea, more than half of them infected at the medical center where the original patient was diagnosed. In South Korea, it is the custom for multiple family members to crowd into a family member's hospital room. It is this overcrowded hospital condition that makes the otherwise modern South Korean hospitals particularly vulnerable to MERS. All those in the country who have the virus were infected in hospitals. Of them, 65 were relatives, friends, or family-hired caretakers who contracted the disease while they were visiting or looking after hospitalized patients. In the same week, a German man who had just returned from Abu Dhabi was diagnosed with MERS, and he died. In Thailand, a 75-year-old man who recently arrived from Oman for treatment of a heart condition was also diagnosed with MERS. Available evidence on genetic sequencing has not identified any significant changes in the viruses obtained, suggesting that this is the same virus. There is person-to-person spread with close contact, but no current evidence of sustained community transmission. A recent meeting of the World Health Organization concluded that the conditions for a public health emergency or of international concern have not yet been met, but that ongoing surveillance is critical. And this is an ongoing epidemic in South Korea, so it's something that we need to keep our eyes on. On this slide, you can see the dramatic increase in cases that occurred beginning in 2014, but with a steep increase in 2015. Now, the next two topics are things that apply to all of these viruses. The next two topics are things that apply to all of these viruses. The first is the Global Outbreak Alert and Response Network. This network, called GOARN, was established in 2000 as a network of international health institutions and organizations willing to contribute and participate in coordinated responses to infectious disease outbreaks. It reflected recognition of the need to strengthen and coordinate rapid mobilization of experts in response to international outbreaks. The network partners agreed that the World Health Organization would coordinate the network and provide a secretariat which would function as the operational support center. The network has evolved to comprise 153 institutions or technical partners and 37 additional networks, the latter encompassing a further 355 members, and has been directly involved in missions to 79 countries, territories, or areas. Over the last 15 years, there has been a continual emergence and resurgence of epidemic-prone infectious diseases and of novel, previously unrecognized emerging diseases that threaten global health and economic stability. In response, GOARN has supported and coordinated the network activities to provide an operational framework for partners to respond to major outbreaks of traditional diseases like cholera and new diseases like SARS and Ebola. Partners have also assisted in addressing the infectious disease impact of major humanitarian crises, including natural disasters. Overall, GOARN partners have been deployed on 137 missions. It is this kind of international cooperation that I think is going to be necessary to bring these kinds of acute viral outbreaks under control. And the final topic is virus hunters. Increasing population densities, global transportation, irrigation, altered farming, and forestry practice and climate change have all combined to produce the environment that has led to these new and virulent viruses. For the last 20 years, teams of individuals funded by governments, research universities, and privately funded organizations have been working to predict and prevent pandemics by identifying new viruses in vulnerable countries in Africa and Asia before they jump to humans. Known as virus hunters, these individuals, who are immunologists, infectious disease specialists, and epidemiologists by training, provide frontline infectious disease surveillance by screening native hunters and their prey for new viruses which have the potential to cause pandemic disease. Simultaneous with the viral surveillance are educational programs directed at eliminating hunting for wild game, or so-called bushmeat. Unfortunately, as the population of Africa and Asia continues to grow and poverty increases, bushmeat is often the only available source of protein, so the educational programs also focus on safe hunting practices, and rural poverty initiatives try to develop protein alternatives. These teams also develop partnerships with local governments, building the infrastructure that can respond rapidly to any new viral threat. The results of high-tech microbiology like this from on-the-ground surveillance are analyzed on the internet to produce interactive, evolving, digital epidemiologic maps that can be used to predict new pandemics as they emerge. Sophisticated programs have been developed to integrate real-time information on emerging diseases and new viruses with geography and travel patterns. This shift to digital epidemiology combines the old shoe leather, on-the-ground techniques implemented by John Snow in his study of cholera with high-level informatics to forecast potential disease outbreaks and prevent future pandemics. With these new viral agents and viral outbreaks, we are seeing the face of epidemics for the future, and organizations like Virus Hunters and GoARN are the face of our current public health response.